All right, so we are going to take some time to take a look at the lab notebook and how exactly to prepare your lab notebook for each week moving forward. So the very first week of lab, you will be doing the microscopy lab and possibly some other preparation type of lab applications and techniques. So the first lab, the microscopy lab, you will be provided with the actual microscopy lab in printed form, which then you can use in class to write down your results, color in your results, and then also complete your study questions on. You'll be submitting your results separately from your study questions in Canvas. Make sure that those photos are color, and you can use that actual document. Starting with Lab 2, which is what I have on the left side of the screen here, starting with Lab 2, this is the first lab that you will need to have prepared in your lab notebook for the next week. So I'd like to show you exactly how to do that. On the left-hand side here, we have Lab 2, the first part of it, and then what we have over here is our lab notebook guidelines. So you should have already gone through all of this information, and you can read through this on your own. What I'd like to discuss specifically in this video is using the notebook prior to lab. So again, each week you will be assigned to prepare a lab in your lab notebook. What we're going over is what needs to be prepared. So let's say this next week you have Lab 2 due. So you need to prepare Lab 2 in your lab notebook. There are multiple reasons for this. One reason is so that we know ahead of time that you have already read through the lab. So you know exactly what you're going into. You know the background information, you understand what you're doing, you're understanding why you're doing it, and you even know the procedure. You've looked at the procedure, you know the procedure, so that when you get in there, you are not confused by what you see before you. You're not confused by the different things that are in the lab room. You're not, confused about what you should be doing or shouldn't be doing. You've already read the information and you know exactly what you're doing. A second reason is because at the end of the semester, you're going to have your lab unknowns. Your unknown project is just that. You're going to be given an agar plate and a broth, and you're going to have to identify what that is. So your lab notebook is what you will be taking with you into the classroom when you're doing your unknowns. So anything that you write down in your lab notebook for how to do a procedure, any notes, anything, is what you'll have available to you when you are doing your unknowns. You will not be allowed to have any other devices, any other papers, anything other than your lab notebook and a pen. So it's important that you have all of the procedures written down that you may need going into your unknown. So that's why this is incredibly important. Also, just week to week, your lab notebook is what you will be using when you're doing your actual labs. So in the actual lab room for Lab 2, for example, you'll be following in your lab notebook, and you'll need to know what to expect and how to do that. So let's get into it. Using the notebook prior to lab, I'm reading over here. You'll be provided with electronic versions or print versions. So I have the electronic versions posted on Canvas. Prior to class, you'll be using that, and you will need to read the lab materials and then write out the following. So this is in the exact order shown. And it's important that each week you do this correctly because you will be signed off on your lab notebook. And then at the end of the semester, your 100 points that go to your lab notebook are all going to be based on your unknown, as well as that you have completed these things each week. So you have signatures and dates for each of these things that you've prepared ahead of time. So here we are. The very first thing that you need to write in your lab notebook for a particular lab each week is on the very first line, you write the lab number and the title. And then later on, you'll write the date that you completed it. So up here, this is lab number 2, and then this is the title, Staining of Bacterial Specimens Part 1. Now I'm going to show you another student's lab notebook for reference. However, if you copy what this student has, that will be plagiarism and you'll get a 0 on your assignment. And I don't think you want a 0 on a 100-point assignment. So please, let's make sure that we are not plagiarizing another student's, but I want to show you an example. So the other student's example here. So this is page number 1. The lab 2 is the first one that you'll be writing up. And you'll notice on the very first line, it says Lab 2, Staining of Bacterial Specimens, Part 1, just like we said in the guidelines. The second line is the subtitle or section or part of the lab, if applicable, and it is in Lab 2. So if we come on over here we see that the very first part of this lab is the preparation of bacterial specimens. So on line 2, you're going to be putting exactly that. And you'll see in this one right here, line 2, preparation of bacterial specimens. Then if we go back to our guidelines on line 3, starting in line 3, it's our introduction or description of the lab work to be done. This is not copying what you have been given, so not copying all of this information here that we see on the left-hand side of the screen. What you need to do is read all of this information, and then what you need to do is take all that information in your brain and write it down in your own words. So again, this is a plagiarism point here, that you write it down in your own words. Now, this is just going to be a little background section. It can be just a couple of sentences. If you're a person that likes to write out more, maybe it'll be more than a couple of sentences. But what needs to be addressed in this paragraph is a little bit of background on why we would be doing this. So why we would be preparing bacterial specimens for staining. A little bit of background on what to expect from it as well. And also what one would use it for. So on here it says, in this portion you'll be writing out a little background on why one would be doing the lab work that you're doing, what it is used for, and then you'll also be writing out what you would expect to see if applicable. So we're starting out here with something that doesn't apply to all of these things. However, you'll see here on the example, this is their line 3 where they're starting the paragraph. This is their description. And what they say is the procedures describe the methods of adhering bacterial samples to a glass slide in order to visualize them under a microscope. So again, don't copy that, but that's what this is talking about here. That's what we're doing in preparing bacterial specimens. Then, so in this case, we don't have what we would expect to see, and I'll give you an example of that in just a minute, one that's a little bit more clear when we get to the gram stain. The 4th step is the list of organisms that will be used. In this one, we're just describing how to prepare a bacterial smear. So as in this lab notebook, it says organisms and there are none. So you don't have to have the line that says organisms. She did, and that's fine, but there is nothing there. Then the next one is the stepwise procedure to be completed, and this should be written out very clearly and organized so that you can follow it while you're working in the lab. You will be using this procedure in the lab, so it should be complete. Each step needs to start on a new line, and this is the only part of the lab notebook that you can copy from what you've been provided. Okay? You don't have to try to reword the actual steps, the actual procedure. You can if you want to. Sometimes you might want to shorten it if you kind of get the point. So, for example, this step right here, you might just want to say, place a loop in the bacterial broth and put a loop full of broth on the glass slide. Rub it into a nickel-sized smear. You will probably actually want to write the whole thing. I take that back, because then you're going to want to let it air dry completely. Maybe you want to omit do not use heat to dry your smear. Maybe you already know that and you don't need to write that as part of your procedure, and you can leave that out. Or maybe you don't want to write it every single time because it's down here as well, and you can choose to not do that. However, I am telling you that at this point, this is when you can write these things out word for word if you choose to. So make sure it's each step word for word if you choose to, or your own words, whatever's easier for you. So if you look at my example here, right here, so using a broth culture, step 1, obtain a clean glass slide. Basically, that's what this says. 2, agitate the culture broth tube gently to disperse the bacteria. Right here, gently agitate your culture broth tube to disperse the bacteria. So you can see where we're going with this. We're just doing step by step. Here's the broth culture, which you can see here, which she wrote down 6 steps there. Down here, using an agar plate with the steps for that. Heat fix the slide, the steps for heat fixing, and then she included the optional note down here. Okay. So then in this here, it says draw out any tables or charts that will be completed as part of the results. In this case, we're not talking about results because this is more background, how to prepare something for the actual lab work that you'll be doing next. And we'll go through these steps with that actual lab so you have a better idea of what you'll be doing for every single lab here on out. So you'll draw out any tables or charts as part of the results. We don't have any here. And then you want to leave space to write out any notes that you have while you're working on the lab, and space for those results. Sometimes we will get into the lab and we won't have a particular organism. Maybe it didn't grow very well, so we're not able to use it, and we use a different organism. This space at the end of the lab would be a space where you would make note of that, saying instead of this organism, we use this one. Or this is a space just to jot down notes. Maybe you're taking time and you say, okay, at 2:15, I need to take this off of this, and you want to make a note to yourself. Or something like that would be even more important would be, say, for example, you did a better job making a gram stain with the broth rather than the agar plate. And then you might want to make a note of that, saying it's, you got a better result with the broth rather than the agar plate, or vice versa. So that when you get to the unknowns at the end of the semester, and we go all the way back to the beginning here to things we haven't done in a while, you'll read through that and say, oh yes, I did do a better gram stain with the broth than the agar plate, for example. And then maybe you'll use your unknown broth rather than your unknown agar plate. So this doesn't have to be pretty, it's just what it is, write down in the lab notebook, okay? So let me go back over these here with our actual first kind of full lab portion. So if we go up to the top, we're going to start on a brand new piece of paper, the very next paper. Remember, you do not write on the backs of the paper, it's only the fronts of the paper, and if you make a mistake, you need to put one line through it and then move on. If it's an entire page of mistake, you put a diagonal line through the whole thing and then move on to the next page. So we just finished right here going through the procedure. Now look, here we are on the next page. The next page, lab number and title. We go back to the lab number, which is lab 2 still, and the title is still Staining of Bacterial Samples Part 1, or Bacterial Specimens Part 1. The second line is our subtitle or section or part of the lab. So for example, gram stain, here we are, here's our lab. Gram stain is this subsection to the lab. Part 3, the introduction. Remember, you have different parts of this. You want to give background on it. You also want to say why you would do something like this, because that's going to be important when you get to your unknown. You have your unknown sample in front of you. All you have is your notebook. You're going to want to know why you would go back and do any of these labs. Because if you're in a situation where you're like, I don't know if this is a gram positive or gram negative, and then you look at the gram stain, you say, oh, one of the reasons we would do a gram stain is to determine if it's gram positive or gram negative. Okay, well then I'm going to do a gram stain. So why you would do the lab, but also what's really, really important to put in here is what you expect to see. So let's take a look at that. Over here in the actual lab, the gram stain, it talks about gram negative, gram positive. Again, this is all background information. This is stuff that you're going to use for your summary. Some of the key things here, what to expect is gram positive cells are purple and gram negative cells are pink. That's what we're talking about as far as expectation. We're not talking about if I have this organism here, what do I expect? What I'm saying is, if I'm doing this type of procedure, what would I expect at the end of the procedure? So if we look at the lab notebook, here we are, the description uses a gram stain to discriminate between gram positive and gram negative. That's why we would do it. Gram positive bacteria, because it has a thick layer of peptidoglycan, stains purple. Gram negative, a thinner layer, stains pink. So we're saying why we would do this, what we would expect to see. Gram positive, purple, gram negative, pink. Next one, list of organisms that will be used, if any. Here the organisms are listed out. There was a mistake made here, or more likely what we did was we changed it in class, and you can then do that, strike that out, and then add what we ended up using in class. Next one, stepwise procedure. Each new one on a new line. So here's the procedure. Now you can do this in a lab where you can say see page, whatever, to reference back to another lab. And this will be important toward the end of the semester, because at the end of the semester we'll be doing things like an MSA or a McConkey auger. And so you'll say just reference this page in this lab. So you don't have to write out the steps or the procedure on how to use those materials. You'll just reference where you wrote it the first time. So here for the procedure, you'll notice that it says step 1 is prepare a bacterial smear with all of the 3 organisms. So here, prepare bacterial smear, see page 1 and 2, because that's what they were for her, page 1 and page 2. And then of all 3 organisms listed above. use 1 or 2 loopholes each. And then even in the second step, heat fix the specimen, see page 2. And then continuing on here. So here is going through the entire procedure of how to do a gram stain. So she goes through the entire procedure of how to do a gram stain. Goes through. And then the next one, 6, what you need to prepare ahead of time is drawing out any tables or charts that will be completed as part of your results. So in the actual lab that you're provided, this is the section that you should be then copying into your lab notebook. So you don't need to copy this part using colored pencils, draw and label these things, but what you do need to put is this big circle where you will be drawing and labeling. You also need to put this rectangle because you're going to be placing your slide on your lab notebook to take a picture. And then you need to put your name there because when you submit the results, I won't have your lab notebook and you need to have your name there. So when I'm looking at the images, then I can see whose image it is. So then here, when you are preparing to get into the lab, what you should have in the results section is a big circle here, a rectangle here, and then space for your name here. That's what you need to go into the lab with. So then after that, the next one is space to write down any notes you are working in the lab and space for your results. So here, note that there's space here. She made a note. I may have smeared too much of the bacterium, compared it to other organisms or compared to other ones. She makes notes. So this is her drawing here. I took a black and white image. You need to make sure that you take color images because you're using colored pencils on purpose so that I can see the difference between your gram negative and gram positive. This is because I took this black and white image. So here, she turned it in and it was in color, and then she was able to label the correct things. Label epidermidis, megatherium, and E. coli. The study questions do not go in the lab notebook. Study questions are completely separate from the lab notebook and need to be turned in separately on Canvas, whether or not you want to use a document for it and type it in, whether or not you want to handwrite it in, however that works for you. That's completely separate from the lab notebook and doesn't go in the lab notebook. So there's study questions there. Now, then we have a part 3 technically to this lab. So again, we go back to the beginning, lab number and title. Here we go. Results, right here, lab number, title. Second line, subtitle, capsule stain. Third one, introduction, description. Here's your description written out. Fourth one, organisms. Fifth one, procedure. Going through the procedure. Results. So draw out any tables or charts. Again, we get down to our results. Big circle for this one, a rectangle for this one, and then you're ready to get into the classroom. Then when you're in the classroom, of course, you'll be writing down your results and completing those. Notice that in this lab it says that you need to post to the assignment in Canvas labeled Lab 2 results as one file with the previous results. So for this lab, what needs to be posted to your Lab 2 results by the next week is this section completed in your lab notebook. So including the colored pencil drawing here, your labeling of the different organisms, and your slide being in here. In the lab, you will do all of these things, and then you will take a picture of this. Then you will do the capsule stain. In the capsule stain, you will do this using colored pencils, draw what it looks like, and then put your slide here, and then you will take a picture of this. Now you have 2 pictures. You need to take those 2 pictures, put them together in a Word document or PDF, and submit them to your Lab 2 results assignment. Again, your study questions are separate. They don't go in the lab notebook. However, you need to complete the study questions for the gram stain portion that we just saw, complete the study questions for the capsule stain portion that is right here, take a picture of the gram stain ones, take a picture of these ones, combine them in one Word document or PDF, and then submit them as one file to study questions, Lab 2 study questions. This is what you will be doing for all of the labs. So, the first week or 2, it is a little tedious. It's a little bit confusing, trying to figure out what goes on what line. I hope this was helpful. I promise you that as we get into the semester, as we're even on week 3 and week 4, this will just be second nature. You'll just put the right things on the right lines and it won't even be a big deal. So there's a little bit of a learning curve, but we can all get through it and you'll be just fine. So if you have any questions, feel free to text me on Remind or email me. Let me know if you have any questions or if I didn't go over something very clearly or if you need anything else answered. So, all right, thank you so much.